Award 2003, Udyog Ratna Award 2011 and 2014, etc. Sir, we are honored to have you here on this gracious occasion. Thank you so much. To honor our rapporteurs. Thank you, sir. Sir Gadak sir, now the session is over to you, sir.
Good afternoon to all of you. Friends, today we gather here the technical session first under the national seminar on abiotic stress management for sustainable millet-based production system, that is genetic enhancement and climate smart practice. Under this session, three key speakers will present their presentations. That is Dr. C. Viswanathan, Joint Secretary, ICR, IRA, New Delhi. Second is the Dr. Kuldeep Singh, ICRISAT, Hyderabad. And third is Dr. P.L. Patil, Vice Chancellor of US Dharwad. Then I request to all speakers, they have given about 20 minutes for each presentation lecture, and we request to all of them to within 15 minutes you cover your presentations or lecture, and afterward 5 minutes will give it to the discussion. Thank you very much. One by one we can start now. First is the Dr. C. Viswanathan, Joint Secretary, Joint Director, ICR, IRA, New Delhi. He will deliver the lecture on harnessing genomic approach for improvement of millet productivity under the climate change scenario. And they will start their presentation or lecture. Thank you.
Good afternoon to everyone. Can you load the slides, sir? Respected chair, co-chair, rapporteur, and distinguished delegates of the conference. Good afternoon to all of you. So today I'll be talking about genomics-based improvement of approaches for improving the millets productivity under climate change scenario. So in this session, I'll be mainly talking about one of the approach, that is genome editing approach for fast tracking the genetic improvement of the millet productivity and climate resilience. My subsequent speakers, they will cover all the other area. So I am from the Indian Agricultural Research Institute. Our institute is, as most of you know, that it is one of the premier institute of the Indian agriculture since 1905. It is dedicated its service to the nation, and we have produced 4 World Food Laureates. Our 4 alumni are World Food Laureates. And it is playing very important role. Not only it played in Green Revolution, today also it play very important role. Just one point I have given: the economic surplus generated by the Pusa varieties is 133,000 crores for the last 10 years, excluding other technologies. So today I'll be talking about the genome editing for fast tracking genetic improvement. This is one of the genome aided breeding approaches. So we know that we need to increase the food grain production to feed the population by 9.7 billion population by 2050 with the existing land and other resources that we have. And we also know that currently we are deficient in micronutrient in our foods produced. So what is the substitute? That is one important aspect that this conference is addressing, and we are thinking that the millets can be one important food grain that can alleviate the micronutrient malnourishment. So this is just another example showing how many people are undernourished people live in India. So this I not explain. So in this conference, we are also talking about how the millets can be improved for climate resilient. As many of us know that the millets are climate resilient, and they use less water and less nutrient as compared to other crops. But we also think that in future climate change scenario, how they are going to perform. And although the water requirement currently it may be less, at least for rice, we know that the water requirement, it will be going to increase more than 15% in future climate change scenario for the same amount of the food grain produced. So in this context, if we can shift to the crops that are less water required, it will be very useful. So this is the demand, like for Nutri-Core cereal, including the millets. We need to produce about 74 million tonnes to meet our the demand for the 2050. And this millets are not new to us. This is from 3000 BC was the first we got evidence that we grow millet in India. And the millets are grown in 131 countries, and about 59 crore people in Asia and Africa, they use it as traditional food. And this morning, our DDG told that how the International Year of Millet was established. So if we see the production area and yield of the millets in India, as told in the morning inaugural session also, in 1950s, our area was around 35 million hectare, and the production was about 15 million tonnes. And today the area is about 17 million tonnes, and we are producing about 14 million hectare, and we are producing about 17 million tonnes of the millets, and we can see that this achieved mainly because of the improvement in the productivity per se. Whether we can increase the productivity over and above, that is the major question, that one of the question that India is going to address, or ICR is going to address. So India contributes to about 20% of the area occupied by the millets, and the in terms of production, it contributes about 20% of the production. But if you see the productivity, it is less as compared to America. Maybe it depends upon the millet that we grow and also the production conditions. So our productivity is about 1.2 tons. And again, if you see millet wise, if you see the difference, it ranges from 0.85 to 1.43 tons per hectare. That is the productivity. So if we compare this productivity with wheat and rice, obviously the wheat and rice have significantly higher productivity. So if you want to enhance the productivity of the millet, we need to enhance the productivity of the millets for popularizing it. So towards this, ICR has released a large number of varieties. This is in the North system. Almost in the last 10 years, about 154 varieties have been released. We need to do more for releasing more varieties that are climate resilient and more productive. This is one of the analysis done by IRI, which shows that in Bajra we are surplus. That is by 2029 we will be having about 3.5 million tons of Bajra that can be exported. But in Jowar and Ragi, we will be in deficit, and in small millets we will have little to export. So we need to produce more in a climate resilient way. And this is some statistics that shows that the global millet market is going to increase to about 18.3 billion. And in addition to that, the millets that are required for distilleries are also going to increase. That is not only for the food consumption, but also for the distillery purpose, the millet requirement is going to increase. If we need to increase the productivity, we need to increase the genetic gain. So in the genetic gain, there are various components out there. That is the selection intensity, selection accuracy, the genetic variation, and various points we need to increase while reducing the generation time. And also we need to see how to increase the genetic gain without bringing in the inbreeding depression or the linkage tract that is common in breeding program. So various approaches that are available is use of the available germplasm and genomic assisted breeding, which will be talked later in some of the session, and phenomics also Dr. Rane will cover. And speed breeding, double diploid, various approaches are there. Genome editing is one of the important approach that we can use to quickly improve the stress tolerance and the productivity in any crop, including millets. So for genome editing, we need the one important thing. What we need is the genomic resources. In millets, it is available now. Most of the millets, the genome sequence is available, and although the genes are not characterized well, but we know how many genes it contain, what are the genes are there. So based on this, we can do genome editing based on the approach that we suggest later in the presentation. By using the genomic assisted breeding, there are several approaches are there, and these approaches I am sure it will be covered in the later presentation by using either the genome association studies or the QTL based studies. We can map the genes and then utilize the genes for increasing the productivity, or we can use genomic selection to improve the productivity, or a mutational approach conventionally used was tilling that can also be used for improving the productivity. But today we will see a targeted approach to generate mutation and use it for the breeding purpose. So everybody knows that the mutation are the source of genetic variation, and that is the source of the one important trait that is required for the crop improvement. And the power of mutation also we know. These are the two mutations. One is the reduced height one mutation that is in the wheat. Here we can see Norman Borlaug standing in the wheat field, which he used the reduced height one gene, which he put in this tall variety and made it dwarf. And this is the SD1 mutation, semi-dwarf1 mutation, which was from the DG gene. It was transferred to the PETA, and then the IR8, the wonder rice variety, was developed. So if we say that these two, the spontaneous mutation that saved the world from starvation, it is not an exaggeration. So that is the power of mutation. Can we generate a desired mutation? Can we use it in the breeding program? That is the major question. And this question was asked whether we can induce the mutation, and this was also well established long back. And Stadler and Muller, they have shown that the mutation can be induced in flies as well as in maize. But these mutations are random, unpredictable, and rare with respect to the gene of our interest. But if you see all the basis of these mutation, all these mutagenic agents, they create DNA damage. And once the DNA damage is repaired by natural mechanism, the mutations arise because of the repair mechanism is not 100% foolproof. So if we can generate this DNA damage at specific targeted location, then we can generate mutation at specific targeted location. So that is the basic based on which the entire genome editing technology is worked. So we give the reagent into the cell so that the DNA damage that occurs in specific place, and then the mutation is created. So this technology was discovered by Jennifer and Dorna, and they used an RNA-guided genome editing approach, which I will describe in the next slide, for which they have been awarded with Nobel Prize in 2020. So this technology is very simple technology. We use two reagents. One is the guide RNA, which takes the molecular scissors to the DNA of the location in which we are interested. As I told, we need to create a DNA damage at specific location so that mutation can be created. So we need a molecular scissor and guide to take the molecular scissor to that specific location. So these are need to be delivered into the cell. It can be delivered as DNA, or it can be delivered as RNA and protein. Various delivery mechanisms are there. It can be aerobic bacteria mediated or particle bombardment mediated genetic transmission, or it can be the protoplast transfection approach. So I will not be able to describe in detail how these methods. So briefly, once these reagents are inside, they go to the specific location in the genome, and they create a double-stranded break in the genome. So once the double-stranded break is created, this can be repaired by three different ways. One is, after making the double-stranded break, we will leave it to the cell that the natural non-homologous enjoining mechanism operates, and the mutation is this place is repaired. During this process, some error can occur that leads to the SDM1 type of mutation. Or if we provide a repair template, that is a DNA template which is similar to that of the native gene, except for one or two amino acid change that we are intended to make, then we can create the SDN2 type of mutation. And we can also insert a foreign gene or a native gene into a specific location. That is done by the SDN3 approach, wherein the gene of our interest is flanked by the location where we create double-stranded repair. So the homologous region is there from the cut site, and we put the foreign gene in between. So based on this, we can create this mutation. And there are several advancements are there. This include the base editing, where only we can change any specific base. We can convert C to T or C to G. Or we can also use prime editing approach, wherein we need not provide exogenous template till we can alter the sequence. So based on these approaches, we can develop allied cultivars. We can improve the cultivar genetic defect. We can introduce stress tolerance, and various approaches can be done. So in general, when we talk of the genome editing, we mainly think that only knockout can be created. So depending upon the location in which we target the guide RNA and the approach we use, we can either knock out or we can alter the expression. That is, we can make the gene to express only in certain condition and not to express in other condition. Or we can increase the activity of the gene by removing the URF. Or we can alter the microRNA binding site so that we can increase the transcript profile. So these are various approaches are available. If some of the students are interested, we can discuss later after the presentation. So once this technology came in, since 2013, large number of people are working. Almost about in 2020, about 231 products have been developed worldwide. Although India is lagging behind, soon we will be able to catch up. And if you see the list, millets are not figuring in. So maybe in a year or two we can see few millets. Like recently, few millets have been edited. Those example I will be presenting to you. And this genome edited product, they are not GM crop. That's why this is advantage over GM technology, as GM technology is not accepted by many people, although there is no problem with that technology. So in case of genome editing, the transgene and the mutation, they are segregated during the breeding process. So finally we get a mutant which is indistinguishable from the conventional mutagenesis process. So that's why it is called advanced breeding tool, advanced mutagenesis tool. Without introducing foreign DNA, also we can produce genome editing, wherein we use the guide RNA and the Cas9 protein. We can make a complex of ribonucleoprotein complex, and this complex can be delivered into protoplast, which can be regenerated to produce mutant. Can you move the next slide? Yeah, previous one. So this technology is now accepted as normal mutation breeding technology or non-GM technology, and many countries in the world accepted this as the technology that is exempted from the normal regulation, GMO regulation that is used in their respective country, including India. So these are some of the products that already they are available in the market. One is the genome edited soybean, which is rich in oleic acid, and the GABA rich tomato, which is good for heart, and also the fishes that can produce more with less feed and within the less time, which can produce more. So these are already example available, and they are already marketed to the consumers. In India also, seeing the potential of this technology, the government has given exemption to the product that are developed from the genome editing technology as per this OM. If the product developed that is free from the introduced exogenous DNA, that will be exempted from the rules 7 to 11 of the rules 1989, that is GM regulation rule. So the DBT has published the guidelines as well as standard operating protocol, how we should proceed about this. So motivated by this, several researchers took up research in the area of genome editing. I will give one example of the line that we developed in the rice, which can be easily extended to other cereals because this gene is conserved. So this is drought and salt tolerant gene, which is present in the rice. We used 2 different guide RNAs to develop different mutant lines. And this shows how the mutant protein, they are differing from the wild type protein. And this mutant, they provide better tolerance to salt stress and better tolerance to drought stress. And since this gene is highly conserved in other cereals, also we can edit in millets wherever it is located, either in bajra or sorghum. In other crops, it can be edited to improve the stress tolerance. So when we improve the stress tolerance, we also need to improve the productivity. That is also one of our objectivity. So in the normal condition, when the stress is not there, the line that are produced should produce more grains. So these mutants that we have developed, all these mutants, they produce more grain in the normal condition. So this is in the Kharif 21, and again in Kharif 22 also we have tested in the field condition in the replicated trial, transgenic field condition. And then again we assessed their drought tolerance in the summer, during this summer, and we can see that in the field drought stress condition also, these 2 mutants, they produce significantly higher grain yield. So this is in the normal yield condition. So if we simply, if we make one mutation in the gene of our interest, we can improve stress tolerance as well as the productivity simultaneously. So this mutant is already in the All India Coordinated Rice Improvement Program field trial. Hope soon we will get results from this one. So similar approach can be used in the millets. So recently, Government of India has approved 500 crores for the millet improvement. Under this program, these are the millets that we have taken for genome editing: pearl millet, sorghum, finger millet, and foxtail millet. Since the genes are not known, and the protocol also not available with our laboratory, but based on the publication, we found that the protocols are there, so we can do it. So we have selected these millets, and the genes are not well characterized. Genes are not available in the. Can you move the slide? Next slide. So in this, what approach that we are using is some of the genes that are highly conserved, that is plant hormone pathway genes are conserved across the plant species. So in one species, if it is established, you can try in other species. Several genes that are related to plant architecture, domestication, they are well characterized in rice. Those genes can be attempted in millets, and some of the genes for primary metabolism, that is in the bajra, to reduce the rancidity, we intend to target the triacylglycerol lipases, which is causing the rancidity. And if we want to alter the amylose-amylopectin ratio, we can target the genes that are present in the starch biosynthesis pathway. And similarly, for herbicide tolerance, we have selected the ALS and ESPS under this project. So for improving the nutrient availability, although the millets are high in micronutrient, the availability is limited by the presence of low phytic acid. So we have selected the ABC transporter for CRISPR-Cas mediated editing, so that the phytic acid content is less in the grain, and thereby the bioavailability of iron and zinc will be higher. Similarly, for although this we are not working on, but this one can easily edit the millets, especially sorghum, for improving the fodder quality by reducing the lignin content. This will not only improve the fodder quality, but in case of if it is used as lignocellulosic input for the ethanol production, it can improve the ethanol content. The genes are available. So many genes, just an example I am showing, if need not worry that in millets the genes are not known, so we cannot start the genome editing program. It is not like that. So several genes are conserved in cereals. This is one of the example. It is in the branched one, which was isolated from the maize, and it has been shown that in the rice, if you edit this gene, it produces large number of tillers. And this is in the switchgrass. They have again shown that if you edit, it can produce large number of tillers. So this function is conserved across the species. So those kind of genes that we can select. And this is another gene involved in the seed size control. Again, if we edit, we can alter the seed size. This has been shown in the different millets, rice, and wheat. And also for shattering resistant, the gene is highly conserved. This is in the rice and foxtail millet, and in the sorghum. The same gene can work across the millet species. So we can select such genes, and we can start the genome editing program in the millets. And again, the herbicide tolerant gene, that is astrolactate synthase, that I have shown, told, it can provide tolerance to rice as well as it can provide tolerance to foxtail millet. So if we select the target genes and create mutation, we will be able to use this technology effectively in producing genome edited plants in the millet crop. For improving the stress tolerance, although the genes, many genes are not known except the DST gene that I have told. In addition to that, we can use the protein phosphatase 2C because these are ABO signaling pathway genes, and this pathway is highly conserved across the plant. So if we create any mutation in the protein phosphatase 2C, the plant will be more stress tolerant and more productive. So these are in Arabidopsis, it has been shown that if we mutate two of the gene, we can produce a better plant and a better stress tolerant plant. So we have used the same approach to edit rice crop. We have developed mutant for, out of the 9 genes, we have developed mutant for about 6 genes. We have already developed mutant, and some of the mutant that we have tested in the laboratory condition, it shows that this can impart stress tolerance. And we can see that the wild type plant almost it dies, whereas the mutant plants they can survive under the severe stress condition. Under normal condition, the productivity is not compromised. Similarly, in the pot culture condition as well as in the field condition also, like the mutant plant, they to conserve water, they roll the leaf early, and they lose less water as compared to that of the wild type plant in the various the in vitro assay procedure. So this suggests that these genes, that is highly conserved genes, that can be taken for genome editing in millet, and that can be used for the genome editing, since till we discover more and more genes by using the genomic information from the millets. So to conclude, the utilization of the conserved gene for genetic improvement by genome editing, we can take it an important approach or in immediate approach, as we know several genes in rice, more than 4,000 genes are characterized. Those genes can be utilized, their orthologs can be utilized to improve the millet. And also initiation of gene discovery program in millet is very important, although we have sequenced several millets, but the detailed functional characterization of genes are very important, because maybe this is very important for producing millets with more dense in nutrient. And it is very important precision mutagenesis tool, and the example from different crops, it shows that we can improve any trait that we are interested in, crops, fish, pig, and various organisms it can be used. And also we need large number of people working in this particular area, as I have shown in one of the slide, that out of the 231 products that have been market ready in 2021, only none was there from India. So when compared to other countries, only few people are working in India. One of the important current limitation in this one is the patent right, which is with the University of Berkeley or the Broad Institute of MIT. So government is now currently negotiating with them to obtain the license. And if you want to be globally competitive, we need to use the genome editing technology to improve the productivity and the stress tolerance of the millets. So with this brief presentation, I thank the organizer for giving me an opportunity. Since time was less, I was a little bit faster. So if have any questions, I will be happy to take this question. Thank you.
Yeah, very nice presentation made by Dr. Sri Vishnatha. Then topic is open for discussion. For 5 minutes, we'll give you for discussion. Any questions you can ask now. Otherwise, we'll you invite to Dr. Kuldeep Singh, ICRISAT Hyderabad, for their keynote lecture. They are giving lecture on the genetic resource for accelerating breeding of millet productivity in semi-arid topics.
At the onset, my sincere thanks to the organizers for giving me this opportunity to be with you this afternoon. In fact, I missed visiting this institute while I was at NVPGR, but got an opportunity today. Realizing that this audience is a diverse audience, what I did is my presentation is based more on this thing. We are an international public good for conservation, preservation for posterity, and development of market-desired dryland crop varieties. So gene banks are basically the service shops, and I'll be trying to give you more information rather than science, which Dr. Viswanathan has given us really good basic science note right now. And that information will be useful not only for geneticists and breeders, but equally for soil scientists, for physiologists, for nutritionists, for food technologists, all of you. Just one slide for the institute. although most of you may be knowing it, but I know some of the students may not be knowing about the ICRISAT. It's a non-profit, non-political international research institute where we are working in almost 54 countries in Asia and Africa. In Africa, we cover most of the sub-Saharan region, especially in the Eastern and South Africa and Western and Central Africa. And of course, directly working on these 4 SDGs. Well, this is the dilemma which has been demonstrated, well spoken by everyone this morning, especially in the inaugural session. From somewhere 7,000 plants which were used as food at some stage, now almost 60% of the calories we are deriving from these 3 crops. Almost 90% of the calories from around 24 crops or so. So this is definitely something which Dr. Chaudhary spoke so well this morning. I think we don't need to repeat that one because he was quite explicit on that one. And it was based on this one that the global biodiversity outlook stressed as and of course combining that with the COVID-19, the more emphasis has gone to the food systems. And there were quite a good number of the global or international conferences, dialogues, country interventions, where we're talking more and more and more on food systems rather than the individual crops. And here I will come to the genetic resources. If we see globally, 7.4 million samples are conserved in almost 1,750 gene banks worldwide, of which 11% of the germplasm is conserved in the CGIAR institutions. There are 11 gene banks in the CG system. And since our topic is on millets, in fact, there was a good debate globally what to call as millets. There was a contentious issue of whether sorghum should be in millets or not, but India played a very strong point in this one, so that ultimately FAO designated 15 crops as the millets, of which ICRISAT is conserving the genetic resources of 8 millets, including sorghum, pearl millet, foxtail millet, little, kodo, proso, and barnyard, and finger millet. But our national gene bank at NBPGR, in addition to these 8 crops, is also conserving germplasm of job steer, brown top, millet, and a little bit of teff, which was introduced prior to 1993 from Ethiopia. Thereafter, now getting the germplasm from Ethiopia for teff is almost difficult, almost impossible, because they have taken it as a national crop where they're making a lot of economic use, economic value out of that one. As we know that most of these crops which we are growing for quite long, for several thousands of years, but at least 4th of these crops, for example, little millet, it got domesticated in India. Indian barnyard millet got domesticated in India. Kodo millet also got domesticated here in India. All others are really the introductions at the later stage. This is a point which has been strongly dealt this morning. When it comes to sorghum, maybe if the data was presented, let's say 30 or 40 years back, probably India was at #1 in terms of total area and other things, but now it has gone down. But other millets in production, definitely for millets, India is the top. But one concern, though, we do say that area has gone down. Production has remained more or less the same, though yields have gone up. But if we see the production 30 or 40 years back, it's same today, but our population has gone 3 times. So that means millet consumption for the last 50 to 60 years has gone down by 1/3 average. So that's a point that we really need to keep in mind as well. This is the genetic resources with ICRIS Global, especially on millets. Almost 479,000 accessions of these millets are conserved, with ICRIS conserving almost 17% of the total millets being conserved in different gene banks. With which, of course, sorghum is the largest one. And what Dr. Chaudhary told this morning about Maldandi, ICRIS Gene Bank has 132 accessions or types of Maldandi. In fact, I got interested into that last year. We planted that last year, all the 132 accessions, we generated data on that one. We are replicating it and repeating it this year to see how is it, how different these 132 are. So that later on we can really look for genomics-based identification, how these 132 are. They may be different because they got adapted in different pockets. Even in Maharashtra, maybe it's a small area, you may have small mutations coming in each of those ones. But this is available in gene banks, and it's available in the National Gene Bank as well. And of course, this gives you specifically the millets in each of the individual gene banks. I will not disturb much on this one, but coming to how these are conserved, we do conserve them under two specific conditions. One is at 4 degrees, which is primarily used for distribution, and at minus 20 degrees, where we use it primarily for long term, where at these stages these could be conserved for 40 to 50 years, because most of the crops with ICRIS is managing, we now have the data that conserving these at minus 20, most of the accessions retain their viability up to 40, 45 years and is continuing. CGIAR institutions took a very conscious decision to replicate the genetic resources, because conserving that at one place only will always be a sort of risk, and all CG gene banks are conserving their genetic resources at 2 places. One is at Global Seed Vault in Svalbard at Norway, and of course thereafter we are conserving this in other gene banks globally. So ICRIS has almost 94% germplasm conserved here in Svalbard, and 92% of in 4 different gene banks in the world. It's not only conserving, this shows the distribution over last 50, 45 years, because ICRIS gene banks 45, 46 years old now. And if we see in terms of numbers, of all the 11 crops with which we are working, 8 millets and 3 legumes, 1.6 million samples have been distributed. Of these, if I go specifically to millets, 0.78 million samples have been distributed in 119 countries. And in fact, sorry. Oh, can we go back? Something happened to this one. Yeah, previous. Yeah, previous one. One more previous, still. Yeah. So this is the distribution of the germplasm of these millets state-wise. The gene bank maintains a complete record of this one. So this is only millets right in India, 57. So we have almost 11,000. That means on an average, each sample has gone 3 to 4 times to different institutions in the country. Well, the same representation, how India has benefited from the ICRIS gene bank. The yellow one, the gray one shows the global distribution, while as the orange one shows the distribution in India. On an average, more than 50% of the distribution has gone to different parts of the country in India. And of course, this germplasm has been used different ways, either directly as germplasm or as parents, and a large number of the varieties they have been released. This is the release only in India, globally, of course. These are the varieties which have been released from this germplasm. Looking at the development of these small millets, of course, I'm not talking much on sorghum and pearl millet, which are major crops in the country where we have very strong breeding programs, both at ICRISAT as well as the national systems. But even if we look at the minor millets, there has been a consistent improvement in all of them. It's not that the improvement is not there, though the number of the varieties or the number of people working on these crops is much, much less than working on the major crops. Coming to the research, so that was the part what ICRISAT Gene Bank is conserving and how much it is distributed. This is the information which people can think of how to use, because if you have 42,000 accessions of sorghum, you may not be able to use all 42,000 in one go. But ICRISAT staff, especially Haripad, did a wonderful work for almost 20 years and developed what we call as cores and mini cores. And probably ICRISAT Gene Bank is the one gene bank where germplasm is very well characterized. So we have the core collections in all these crops here available with us. And using these cores, our other germplasm, we have evaluation of the information on most of the crops. I will take one example of finger millet, and this will be true for all other crops as well. For example, this finger millet germplasm has been characterized for all these characters, be it biotic stresses, abiotic stresses, grain, agronomic traits, whatever it is. For biotic stresses, for example, blast, 80 accessions have been identified which are resistant to blast. This sort of information is available at the ICRISAT webpage. So anyone who's working on this finger millet knows that the blast is one of the major diseases, and you can have access to these 80 accessions. Grain nutrients. Finger millet is one of the richest source of calcium, but the variability is from somewhere 3,000 ppm to almost 5,000 ppm or so. So this is one of the richest source of calcium. All the genetic resources, at least if not all of them, because we have 7,500 accessions, but the core has been characterized for this one. And so this is a huge variability coming for this one. For drought, 11 accessions have been identified which are tolerant to drought. Now, depending upon different conditions, drought again is not a single trait. You may have to define drought as per the soil conditions, soil types, as per the environmental conditions, a lot of things which physiologists are really modifying the definitions of drought now very well. So depending upon the conditions, whatever has been worked into this paper, you have 11 accessions which are drought tolerant. For soil salinity, 21 accessions have been identified which are showing tolerance even up to 125 millimolar sodium concentration under the pod conditions. Of course, if these have to be validated, one has to go to the field conditions, but using this basic stock at the moment. These millets are not only for the human food, they are an important part for our livestock as well, because integrated farming is something which is ultimately going to benefit the small farmers. And for millets, if we are really making them successful, we have to look into the fodder quality as well. So we are really making some efforts to look into the fodder quality as well. And we have seen variability for some of the characteristics. And this was done in collaboration with ILRI, because ILRI has an office right at ICRISAT. So that work is going on here as well. Pearl millet, the germplasm which has been characterized, the iron, because we know right now that one of the richest source of iron among all the cereals is the pearl millet, and I will tell you where from this germplasm has come, and that's the advantage of having a free exchange of germplasm that is coming in my other slide. Similarly in sorghum, you have a lot of variation coming for all these traits. This is available in our web page. And Of course, for high iron, this Parvani Shakti has been a selection from this collection, which is of Indian origin. But for pearl millet, which is high iron and high zinc, which was been released as Dhan Shakti in India and Chakki in Africa, the basic germplasm has come from Togo region in West Africa. So that's the advantage of the global gene banks, where you will have the germplasm from across the world, and that is freely available to anyone working on these crops. So this just gives you, for example, how the millets are important. Neither wheat nor rice can compete these crops when it comes to protein content. We have been talking a lot about iron and zinc, ignoring protein, while we have a national data where 45% of our children below the age of 5 are protein malnourished. So if we really have to improve the protein nourishment, the millets are a good choice for us. And of course, as Dr. Pathak, the Director General, told this morning, that they cannot be commercially viable unless we see that they are economical to the farmers. And to make them economical, we really have to increase the productivity of those ones. And that's where we really see a lot of variability is in this one. And ICRISAT is now really stressing more and more on characterizing these genetic resources on a larger scale, because all the data I have shown is based on the cores which were easy to handle, smaller, with less budget, other things, but now we can do it in the larger scale. I will not go into details of that one because Dr. Vishwanathan has already, sorry. Oh. Yeah. Dr. Vishwanathan has already very well specified the genomics, the genome sequencing of most of these crops is available. But Dr. Vishwanathan, what we see, the trait for which we don't have the variability for is the grain size. And you know there's a good linearity in centennial, rice, and these crops. And in fact, one of the best approach are the successful, most are the required trait for which we need to work is the grain size. If we can improve the grain size of these crops by 15–20%, that can translate directly into the productivity. This, I thank you all and many supporters, because ICRISAT has been supported by FAO, by the Crop Trust, by Plant Treaty, the Government of India. We are planting we have already planted 5,000 accessions of small millets. And anyone who is interested to visit ICRISAT will be organizing field week. We are not organizing a field day because then it becomes restricted to the individuals. We are keeping one full week for this visit. So, and we'll be announcing those through ICRIP. Whosoever is interested can really visit. And this is under Bihar project, because government of Bihar has developing a center of excellence in millets, where they have asked ICRISAT to partner with that one. And under that support project, we are growing all these 5,000 accessions in one go for all these millets. Thank you. Thank you very much.
Thank you, Dr. Kuldeep Singh. Now he has given very good lecture for on behalf of ICRISAT, how to store the millet germplasm and evaluation of millet germplasm. And I request to house any suggestion or any point for discussion. Please take interest for the discussion now. Yes.
Hello, Dr. Kuldeep. Very nice presentation. I was just curious to know whether we can store the germplasm at in liquid nitrogen. You mentioned about 4 degrees Celsius, then minus 20 degrees Celsius. So can we go below that so that we can increase the time period of the germplasm?
Yes, we can do that one. In fact, NBPGR has a cryopreservation unit in it, where most of those crops where seeds don't remain viable after you're drying them, they are being conserved under liquid nitrogen. And second thing which I initiated while I was a director there is getting a good quantity of seeds of the wild species is difficult. And we initiated a program of really conserving the vines. It's going very well, yes. And there's a global interest also in conserving them in liquid nitrogen.
Okay,
Thank you, sir.
next. Yes, Dr. Javade.
Sir, myself, Dr. William Gavle, working as a officer in Infrastructure Organization Station, VNMK, Parvani. Sir, Parvani Shakti variety, it is released in collaboration with ICRISAT and VNMK, Parvani. The same variety also has been released during the last joint agroscope for Rabi season also. Basically, it was previously released for Kharif season, but in Kharif season, there may be the deterioration of grains due to the late rains. The same variety also has been released for Rabi season, which contain good amount of iron and zinc. This is my submission. Thank you.
Okay, anybody now? Yeah, please.
Select characterizing or identification of germplasm from whole complete collection of gene bank is a very Herculean task. Is there any effort to utilize the concept of focused identification of germplasm strategy in ICRISAT? Do you following any this strategy in identification of trait specific germplasm?
Yeah. See, focused the figs
Yes.
is more useful where we are now going for the collection purposes, right? With the plant area, with the CBD and other things, our role in collections has become limited, especially in India. We do not do it. Whenever we do it, it's in collaboration with the NBPGR, and where we are helping them in identifying the missing gaps. But our presence in Africa is pretty very strong because as of today, whole of the Africa region has given equal opportunity to ICRISAT for collection and conservation. And there we are actually looking based on the climatic conditions, where those climatic conditions or soil types or other things are present. For example, a lot of area in Africa, especially East Africa, is acidic. And if we have to improve the productivity in Eastern Africa, we really need the acid tolerant germplasm. And that's one area where we are focusing now to see, to identify the regions where you have the acidic soils, and what is germplasm which we have collected from those regions, and where are the gaps, so that we can focus on that one. Yeah.
Then any other from house? Then our co-chair, Dr. Praveen Kumar, are you want to share anything?
First of all, I would like to compliment Dr. Viswanathan and Dr. Kuldeep Singh for the very explicit and wonderful presentations. And as told by our honorable DG sir, that since in last 75 years, if you look at the population in last 75 years, population has increased by 3.9 times. And rice production has increased by 6.6 times. Wheat production has increased by 17.5 times. But if you talk of the millets, its production is increased only by 1.5 times. Then the area has decreased, though productivity has increased, but still productivity is almost at par with the world average. There is a lot of scope, and how that productivity can be increased, and those physiological approaches and has been very, in a very nice way, has been explained by our colleague Dr. Viswanathan, and how much the, what is the status of the global sections available in the ICRISAT. Of almost 17% of the global genes are available for the millets in the with the research. So there is a lot of scope that that biotic stress and abiotic stress which are bothering, which are the bottleneck in increasing the production of these millets can be addressed to a great extent. And this is I would like to share my personal experience also. Last like 8-9 months, that millets is the integrated part of my diet, and I have found that I could able to reduce 8 kg weight, and almost I could reverse my diabetes in last 9 months. So millets has a great potential. The main advantage is, which I could able to feel, that in rice there is a carbohydrate of 8%. In wheat, the carbohydrate is about 66%. In millets, the carbohydrate is 60 to 65%. So if you taking the low carb diet, then automatically your weight will go reduced. Moreover, it has low GI index. Almost all minor millets has low GI, lesser than the 55 GI. Sorghum and bajra has the medium GI. On the other hand, the ragi is only high GI. But calcium content, those people who are not diabetic, but calcium content in ragi, in finger millet, is 5,000 times than the rice. And Dr. Vishwanathan was referring that the GABA in tomatoes. If you talk about the GABA, that is lecithin fatty acids, it is abundantly available in the almost all millets. In other cereals, it is not available, and that is very useful for your nervous systems. So regarding water requirement, the water requirements of most of the millets is 2.5 times less than the rice. And water is a very scarce commodity. So in future, there is urgent need to shift some of the area under millets, and it has lot of its nutritional profile is excellent, and moreover, it has a very high satiety index. If you are taking the same satisfaction, if you are taking by 150 gram of rice, the same satisfaction you can derive by taking only 100 gram of carbohydrates. That those millets, if you are taking 100 gram millets, the same satisfaction you will get which you are getting by taking 150 gram of rice. Moreover, carbohydrate is less, quantity you are taking is less. That is with low GI food, that is ultimately helping you to get your good health, and it is also for the healthy heart also. Since most of the millets, it is loaded with magnesium, which is very good for hearts. Similarly, all of the minor millets have alkaline foods. They don't form the acid in your body, and that alkalinity help you to keep your heart healthy. It will avoid you, आपको stroke नहीं होगा if you are taking the alkaline food. So there are a lot of nutritional advantages. Since last 9 months I am taking millets, so in depth I am studying that, and this was all these two both the talks were wonderful, and thank you very much. Now over to my chairman.
Okay, thank you, Praveen Kumar. Now, any suggestion or any questions from house? Okay.
Good afternoon, everybody. I am Santosh from Krida. Sir, my question is to Dr. Chinnaswamy Vishwanathan, sir. Sir, thank you for the wonderful presentation. I just want to know, like recently government has legally mandated the restricted use of glyphosate. Some of us were earlier working, and few of us are continue to work on EPSPS. So is it prudent on our part to continue that targeting that gene? That's one question. Second, if I heard you properly, sir, you said the genome-edited mutant lines have yielded more grains in normal condition. So I'm just curious how a drought-tolerant line having its own metabolic cost can yield better in the normal condition. Just two questions, sir. Thank you.
So thank you. On the first one is on the glyphosate restricted uses. I know that the government has restricted the use of glyphosate because of some reports in USA that it causes cancer, and although scientifically not proven, because of various reasons, the government has restricted its use. And this is one of the herbicide that can be used, like it broad spectrum herbicide. That's why we are working on it, and we are also introgressing this transgenic version of the glyphosate resistance in the ESPS gene in the soybean. Already almost 7 varieties are almost ready. So if government permits, that will be released soon. But we are not sure what the stand government will take. So the alternate one is we can use the ALS, or we can use any other gene which are safer herbicides are there. We can use the other approach. The second question is on how the DST gene increasing the yield as well as it is increasing the stress tolerance. So the drought and salt tolerance, this is a C2H2 zinc finger transcription factor. It has a lot of target genes. By transcriptome, we have identified almost about 600 target genes are there. So this one of the target is the cytokinin oxidase, which earlier identified as GN1A mutation in rice. So if there is mutation in cytokinin oxidase, the cytokinin accumulation is more. It produces more tillers, more grains per panicle, and it produces more yield. So this cytokinin oxidase is positively regulated by the DST. DST is the transcription factor that upregulates the cytokinin oxidase. Since mutation is there in the DST gene, so it induces the more it reduces the accumulation of cytokinin oxidase. So cytokinin accumulation is more, thereby it is promoting more tiller, more leaf area, although the stomatal conductivity is less for this mutant, because it produces more leaf area and the leaf arrangements are in such a way that the overall whole plant photosynthesis is higher for this mutation as compared to that of the wild type plant. Because of this reason, it produces more grain even under the controlled condition and also under drought condition. Thank you.
Okay, thank you. Then, friends, today there are two presentations were made in this our session first. That is from the Dr. C. Vishnatha and Dr. Kuldeep Singh. You know, we are celebrating the millet years now, International Millet Year, and the area under millet is reducing day by day from 35 million hectares to it reduced about 14 million hectares now. But productivity is increasing. It increased about more than 12 quintals per hectares, and this is a very good sign. But this is a proper year because we declare the International Millet Year. On that basis, we want to increase the area under the millet. There is 8 millet crops, that 2 are major crops and 6 are the minor, small millet, minor millet. And ICRISAT has doing the very good work for conservation, evaluation of the millet, small millet particularly. More than 5,000 association numbers they are of the different small millets. They are evaluated and they are storing, are keeping long-term storage in their gene bank. And that's why it is a very good strength of our country. We can utilize this germplasm for our breeding work and further evaluation also in your area. And it is a good opportunity. And I thanks to both speakers for deliver their very good lectures. And I thanks to all the responders. They given the comments or put up the questions in house. Then I thanks to our co-chairman, Dr. Praveen Kumar. He has given his very good suggestion on this aspect, and also thanks to reporters for taking the care for the posting and writing the posting of this meeting. And I thanks to audience also, all the dignitaries present in the hall, director, Vice Chancellors of the ex-Vice Chancellor, Dr. Dhawan sir, and also all my colleagues. I am not taking on name of all because time is limiting factor. And I thanks to all of you for cooperating me for the chairing of this session. Thank you very much.
Thank you so much, sir. The House will agree with me that both the lecturers were very insightful and wonderful. So as a customary and as a token of our love and affection, I request our chairman, sir, and co-chairman, sir, to felicitate our two wonderful keynote lecturers. First one, Dr. C. Vishwanathan, sir. Sir, please come on the dais for delivering the lecture on harnessing genomics approach with improvement of millet productivity under climate change scenario. I request our director, sir, to come on the dais, please, also to join us. I also request Dr. Kuldeep Singh, sir, from ICRIS at Hyderabad, who delivered the keynote lecture on genetic resources for accelerating breeding of millet productivity in semi-arid tropics. Thank you, sir. We have two wonderful rapporteurs with us for noting down the proceedings. So I request our chairman, co-chairman, sir, to felicitate our rapporteurs. Dr. P.S. Basavaraj, scientist, plant breeding and genetics. And Dr. Vijay Sinha Kakade, sir, scientist, fruit science. So thank you. Now I request our director, sir, Dr. K. Sami Reddy, sir, to felicitate our co-chairman, sir, Dr. Parveen Kumar, Director ICAR CCRI Goa, for his gracious presence on the occasion. Thank you, sir. I also request our director, sir, to felicitate the chairman of this keynote session, Dr. S.R. Gadak, sir, Vice Chancellor, PTKV, Akola. So thank you so much. Now we come to the end of the first keynote session, but before we leave, there are few announcements to make. पोस्टर सेशंस टेक्निकल 1 और 2 के जो पोस्टर सेशंस हैं वो जहां जहां पे डिस्प्ले करना है वो थोड़ा आप लोग लगा लीजिए और अभी हम लंच के लिए ब्रेक करेंगे और लंच के बाद टेक्निकल सेशन 2 विल बी हियर टेक्निकल सेशन 1 and 3 विल बी अपस्टेयर्स जिनके स्टेयरकेस एडमिन बिल्डिंग जहां लिखा है वहां पे है और कोई भी वॉलंटियर यहाँ पे है तो आप पूछ सकते हैं वहां जाने के लिए बाय 3 o'clock थोड़ा क्विकली लंच करके आने के लिए रिक्वेस्ट है क्योंकि ऑलरेडी डिले चल रहे हैं और टेक्निकल सेशन 4 स्कूल ऑफ ड्राउट स्ट्रेस मैनेजमेंट में है वहां पे जाने के लिए थैंक यू सो मच
Okay, okay. Thank you.